Okay, this lecture number 10, it is on DNA. We'll have it in two parts, part 1 and part 2, for easy uploading. So DNA is abbreviation for deoxyribonucleic acid. This is the D, deoxy, ribo, and this is the N, nucleic, and this is the A, acid. So DNA. Deoxyribonucleic acid is a genetic material, of course. It is the genetic material which transfers the information from one generation to the other. Within the genetic material, information is organized as genes. What are the genes? They are short segments of DNA that contain instruction for a specific traits. Instruction, details, information. Okay. So your information is stored in small short segment of DNA. Are the genes, and these genes sitting on certain position on the chromosomes, and the position, each position named locus. Okay. Genes are organized into chromosomes, as I said. Yes, the mechanism by which genetic information is transmitted at both the cellular and organismal level, on the organism as whole, as body, human being, or you are take it as a cellular level, from one generation to the next. Since the information stored in the genes sitting in the chromosomes, you remember how during the cell divisions they will divide it to two daughter cells. So then in the meiosis you reduce it to half in the sperm, half in the ova, then the fertilization. So you are the information transmitted from one generation to the other. Okay. In eukaryotic cells, including those of a human, the majority of DNA is located in the nucleus. So where are this store of the information of your genetic makeup? It is in the gene, in the chromosome, and mainly they are sitting in nucleus of the cell. Okay. A small amount of DNA is also found in the mitochondria. Okay. So some of the DNA, it is mainly the majority in the nucleus. However, some of it in the mitochondria. As the genetic material, DNA must be able to do three things. It should have three functions. Function number one, it could be replicate so that it can be transmitted to the next generation. You can't just have one part of the information and send it to the sperm and just part. These has to be replicated, divided. So you copy it, replicate it, you copy it and send it to the next generation. And that could has to, DNA, they are replicated and they are stored. It could be stored in the genes, and the information could be stored and undergo change, mutation, right? So to provide genetic variability. DNA meets all three of these criteria. Okay, the double helix. DNA molecule is a double helix composed of two strands that spiral about each other. Let me go quick that then till I reach the figure. I like the figures. They will show you, simplify this information could be demonstrated in figure. Each strand is a polynucleotide because it is composed of series of nucleotides Okay, nucleotide. What is a nucleotide? It is a molecule composed of 3 subunits. Phosphoric acid, it's phosphorate, it is P. Pentose sugar, deoxyribose, and nitrogen containing base اللي هي الأدينين. Sorry, I'm talking Arabic now. It is the adenine shorted or abbreviated by A, cytosine C, guanine G, and thymine T. So adenine paired or connected to T and C with G. Okay. Yes, adenine to guanine, cytosine to thymine. Sorry, what is it? Adenine and guanine are purine. Yeah, that's right. I thought they are saying adenine to guanine. What is the rubbish? So adenine and guanine are purine, 2 rings, and cytosine and thymine are pyrimidine, 1 rings. Pyrimidines, 1 ring. Right. So here is the helix. The helix DNA double helix. See, this is the DNA, and when you unwound it, just you separate it, it become like ladder. You got ladder at home, right? So it's actually straight ladders. Okay. Sorry, ladders. The supports are composed of sugar S and phosphate P. This is the support. This arm and that one are the supports of the ladder. Okay, they are composed of sugar and phosphorus. Okay, phosphorate. So sugar, phosphorate, sugar, phosphorate, sugar, and this the same. Okay. The rungs, these they call it rungs. You remember the ladder? Wooden one or metal one, 2 parts, the stand, the support, the backbone, and here is connected by rungs. Rungs are complementary base. The DNA base pair in such a way that the sugar phosphate backbone are oriented in different directions. What does that mean? You see this one? It is end by F prime, while the other one 3 prime. Here is 5 and here is 3. So they are sitting in opposite direction. Okay, oriented in different directions. Here is the C part of the figure. So the DNA strands are anti-parallel. Notice that numbering of the carbon atoms 5 prime and 3 prime in different direction in the deoxyribose. You see. So adenine to thymine, right? Adenine connected to thymine, and here you see upper part is the F. This is the 5 end, and here is the 3 brand. Okay, 5, 3, 3, 5. Okay. So this, look to this, this one is part of here. You see, this is 3 prime end and 5 prime end, and thymine connected to adenine, right? Right, sugar will phosphate, the structure of the DNA sugar. Looking at just one strand of DNA, notice that the phosphate and sugar molecule make up the backbone. This is what we said. But the two strands together, put the two strands together, and DNA resemble ladder. Okay? As we said, the sugar phosphate backbone make up the supports. Okay. And how they are connected was the complementary base pairing. So the rungs of the ladder are the paired bases which are held together by hydrogen bond or bonding, hydrogen bond, each one. So they are connected by hydrogen. Okay, a pairs with T, sorry, A pairs with T. So A with T, forming two hydrogen bonds, and G pairs with C, so guanine with cytosine, forming three hydrogen bonds. So this is 2 and here 3. So this is supposed to be, well, it's not supposed to be, it has to be stronger. It is 3 bond, 3 tie. You are tying them 3 times. So this is more stable than that adenine to thymine. This is called complementary based pairing. Complementary based pairing is important to the function of the DNA. The two strands of DNA anti-parallel. You remember when you said part of it is 5 prime and the opposite of it is 3 prime. So one of them is right side up, appear right side up, and this one upside down. One with 5 and 3 opposite to it. Okay, this is due to the position of carbon molecule in the deoxyribonucleic sugar, and these are what we are talking about, 5 and 3. This is the number of the carbon. This orientation become important when the DNA is replicated. Okay, still 1 or 2 minutes to go. Let me finish this slide. When cell divide, each new cell gets an extra copy of DNA. The process of copying of DNA is called DNA replication. During the synthesis phase of interphase during mitosis, when DNA is replicated, the double strand structure of DNA allows each original strand to serve as template for the formation of the complementary new strands. Don't worry about that. I will explain it in the coming figures. Okay, so you have template and you are building a new strand according to the structure of the template. Of course, it will be opposite of it. Okay, if in your original A's, so at the opposite side will be the T, and C will connect it to the G. Okay, I don't know what the number of slides is, number 10, so about the time to save. Yes, let me save it.